All right, for the biodiversity inquiry, I just wanted to take a few moments for teachers specifically to go over some little hints, tips and tricks about this inquiry, because it's a fun inquiry, but there are a few things you want to be aware of before you go ahead and run this with your students. It is a good idea, as I mentioned in the intro video, to make sure you have gloves that are pretty heavy duty. These are leather gloves. They're a little bit hard to They're obviously larger than your typical garden gloves, but there will be the potential for injury from stickers and prickly things. And this is one of those activities where it's really easy to assign different jobs to different students. If you have multiple students that are focusing on one particular area, the gloves with the leather tend to be the best, and you can have one person picking up the leaf litter and putting it in the sifting box, and then they're sifting it. But then you also have other students who may be doing some written observations. So I always encourage students to go out into the field and have somebody that's kind of the recorder if you have multiple students in a group and they're just taking some observational notes. Also keep in mind, you definitely want to encourage students to go back and think about some controls so that you're not sifting at one site for 30 seconds at a time, but then you go to another site and you're sifting for 45 or 60 seconds, because that could potentially affect the results that you get. A lot of times, especially if I have my students go out to a place that happens to be on campus, many schools have areas where you can find places where there's enough leaf litter on the ground that you can use to sample. I will have the students go out during a previous class before they actually do the collection, just so that they can get an idea of what the surroundings are, kind of like a little mini tour. And it only takes 15 minutes or so. You get to go outside. Hopefully the weather's nice, but it also gives you an idea of where you're going when you actually do the collection. Because when you do the collection, depending on how long you have in your class time, you will need to move it along. And if they don't really know where they're going and they're just seeing the place for the first time, then they're not necessarily going to be as efficient with their time. The other thing to keep in mind, not all schools are located in an area where you can just leave the building and stay on campus and do collections, or even do collections nearby. So there may be a potential where you'll need to find a local forested area, park, state park, some other location that would be suitable for this project, in which case you definitely need to, if you do leave a school property, make sure you've received permission and you've notified the people who are in charge of that particular area about what's happening, when you're going to show up, how many students are going to be there. The idea is that we leave the area as similar as we found it, and hopefully not have too much disturbance. But definitely think about that. Think about health issues. Do you have any students who may be allergic to certain things or have allergies, especially bee stings? It's really easy when you're going outside and spending some time doing these collections to get stung by a bee or something else. And so that's something you'll want to let your school health room or health aide or nurse know about, which students are going out, maybe even sending a permission slip, especially if you're going off campus, which would be required. So just some things to think about as you go to different sites. You want to make sure that you pick a site that actually has something to sift in your sifting box. The leaf litter is where your arthropods are hopefully going to come from, and it could be a combination of things like pine needles, like we found here in Eastern Oregon, leaves, dead leaves that have fallen on the ground, maybe a little bit of dirt. And you can actually create your own sifting boxes. Some schools will use those plastic Rubbermaid containers and cut the bottoms out and put a plastic mesh at the bottom and tape it. This is a really commonly used type of sifting box. These boxes are wine boxes that you can get, I believe, for free if you ask them, from Trader Joe's. They make really good sifting boxes because they're tall enough, but they're also easy enough to cut out a square at the bottom and have students or yourself use masking tape, or especially some type of strong duct tape to tape this on. They do have a maybe 2-3 year life expectancy, depending on what the weather's like when you're out doing the sifting. But this box has probably been used for 2 or 3 years at least and is still in pretty good condition. So I would have 1 box per group that's going out. And I would also make sure you have some type of a container. The Home Depot 5-gallon Rubbermaid containers seem to work the best. They also will sometimes suggest students take out their phone so that they can take pictures of the site. Sometimes when you're going back and maybe doing a conclusion or writing about what the site was like, observations, qualitative observations, you forget about what the weather was like, what the temperature was like, what the wind speed might have been. And those are things you can tell just by taking pictures or even looking up on a weather app while the students are out there working. There are different ways to do these arthropod collections depending on how much time you have. If you use pillowcases to collect your leaf litter samples after they've been sifted, as I mentioned in the intro video, pillowcases are nice because they're breathable. So if you only have time to go outside and collect and then the time is up and you don't have time to identify those arthropods, you could easily put these in the room somewhere safe and come back tomorrow, the next day, and hopefully I would think most of the arthropods that were alive when you collected should still be alive when you look at them the next day. So that is an option. I do think as much as possible we tend to get really better results when you sample and at least try to identify and collect on the same day. The thing to keep in mind with this lab is there is a non-kill version. If you have any students who are sensitive to the issue of insects, arthropods being put into rubbing alcohol and killed, one of the reasons for using the USB microscope camera is that you can physically put this on top of the arthropod and take a photo of it. Sometimes it's difficult if they're really small or if they're moving really quickly, which is why we use the rubbing alcohol and the little well plates to do our collections and kind of final photos. But it is possible if you're really careful and have students practice taking pictures with these microscope cameras. They're relatively inexpensive and they just hook up to a Chromebook or a laptop computer. You can take a picture of the arthropod, or you could even, if it's big enough, have them take a picture of it in the field with their phone and then release them back out into the area where you originally collected them. So just keep in mind, it's in the materials on the Patterns website, but there is a non-kill version that is available for use if you have students who are sensitive to that. If you do use rubbing alcohol, I would suggest using some goggles, at least for the time you're using that. This is really the only chemical in this particular inquiry. And then when the students are looking at the different insects, you want to go ahead and make sure that everything has a photo. And so you're actually giving students a chance to look at a photo and potentially identify the species correctly. One thing that I would suggest, it's a little bit easier if you're having trouble. There are websites, I believe, that you could take a picture of the arthropod, submit the picture, and it will, as closely as possible, identify or give you the most likely species that would be. There are also field guides like paper books that you could use depending on your area and what you want your students to do. So if you don't want to use a USB microscope camera, the other option would be dissecting scopes. So, and you can, I know a lot of schools already have these, so you could put the arthropods in well plates and actually put them under the dissecting microscope and look at it that way, especially if it's really small. But hopefully you'll have a successful experience with your students for this inquiry. It is one of those things where we don't always get the results that we expect to get, or maybe even as many bugs or arthropods, but a lot of the focus for this inquiry is on the procedure and the experience of going outside in the field and doing these collections and kind of putting on our little ecologist hat and trying to figure out what are some of the things that might affect biodiversity.